Talking about CRISPR. What does CRISPR even mean though? It stands for the clustered, regularly interspaced, short palindromic repeats. For now, that's all that it means, but we'll explain more about what it stands for later.
But before we dive into the main components of CRISPR, it's important that we get some background information. So first, in 1987, a scientist named Yoshizumi Ishino discovered something strange in E. coli DNA. He noticed short sequences that kept repeating, with unique spacer DNA in between each repeat. At the time, no one knew what this pattern meant, so it was just recorded as an unusual observation. But this repeat spacer repeat structure would later become known as CRISPR, and it turned out to be part of a bacterial immune system. Furthermore, in 2005, scientists discovered that those spacer sequences actually match DNA from viruses that infect bacteria. This showed that bacteria store pieces of viral DNA as a memory of past infections, so when the same virus attacks again, the bacteria can recognize and defend against it. This was the real first evidence that CRISPR works as an adaptive immune system. Then, in 2012, Jennifer Doudna and Emmanuel Charpentier discovered how to turn CRISPR into a precise gene editing tool. They found that a protein called CAS-9 acts like molecular scissors and can be guided by a piece of RNA to a specific DNA sequence. Once it reaches that target, Cas9 can cut the DNA at the exact location. This was a breakthrough because it allowed scientists to edit genes accurately and efficiently. It transformed CRISPR into a natural bacterial defense system, into a powerful tool used in genetics, medicine, and biotechnology. But more on that later. Moving on, in 2013, scientists at Broad Institute and Harvard University successfully used CRISPR-Cas9 to edit DNA in human cells in the lab. This proved that the system could work beyond bacteria, and it was the first step towards using CRISPR in medicine. Now, with all that research, in 2020, Jennifer Doudna and Emmanuel Charpentier won the Nobel Prize in Chemistry for developing CRISPR-Cas9. This is recognized as a huge impact as a gene editing tool. Go women! And by then, CRISPR-Cas9 was globally recognized as a powerful gene editing tool. It's now used in research, medicine, and has already led to treatments for some genetic diseases. Now, enough of that boring history stuff. Let's talk about how CRISPR is used in prokaryotes.
When viral DNA enters bacteria, it becomes a standoff. Viral DNA versus the bacterial immune system. So, let's look at the key players. Players 1 and 2 are Cas1 and Cas2. They are individual components, they work together. And then you have Cas9, which is the final, lasting bacterial defense against the viral DNA. So, let's see what happens. Okay, here's the viral strand. And oh look, there you have Cas1 and Cas2. But what are they doing? They're looking for the PAM site. But what is that? Yo, why is Christopher such a bad barber?
Why?
Because it only cuts when it sees the payment. The payment. Get it?
LOL. Barbershop haircut costs a quarter. Okay, wait, but seriously though, what is PAM?
Okay, yeah, but in all seriousness, the PAM site is where all the action happens. It stands for the protospace adjacent motif. Well, what's the function of the PAM site? Well, it's essential for the CRISPR system to cut and cleave DNA. But where do we find it? Well, let's look at some DNA right now. You have your 3' and your 5', and you're looking for 3 nucleotides. In this case, we've given the example of N followed by 2 Gs, where N means any nucleotide. So, looking for any nucleotide followed by 2 Gs, or guanines. Okay, let's look back at our viral DNA. Here we have the strand, plus the Cas1 and the Cas2 from earlier. But as you can see here, we've introduced a new variable, being the PAM site. In this case, they found the nucleotides A, G, and G, which, if you remember from earlier, is a PAM site. So they proceed to cut that strand of DNA until the PAM site. Alright, so now that we have a cut piece of DNA from the viral DNA, what do we call it? Well, they're called a protospacer. Why? You'll see in a second. Let's look at the bacteria for a second. Here we have something called Crispr RNA, which consists of spacers and repeats, and here we have our protospacer from earlier. What we see now is how this protospacer joins Crispr RNA and becomes part of it, hence becoming a spacer and being part of the CRRNA. Now that we've discussed Cas-1 and Cas-2, let's talk about Cas-9. So here we have our CRRNA, and we're focusing on a spacer and a repeat. This pairing here is really important because it's the structure they make within the Cas-9 itself. So, as you can see, the spacer and the repeat form a bond together and make a shape much like the one you see on screen, forming what we call tracer RNA. This is really important to the structure of Cas9. Let's take a look at our tracer RNA once again. As a reminder, it consists of viral DNA from the protospacer from earlier. This taken forward, it shows us how important it is within the structure of the Cas9. Let's have a look at it. So here we split into two main sections. The second section here in green talks about how it's able to analyze and cut DNA strands from viral DNA. But how is this possible? Taking a closer look, we can see here that tracer RNA is embedded within the structure of Cas9. In this state, it's known as guide RNA or gRNA. This means that when referencing or cross-referencing with viral DNA, it's able to detect which ones have already infected the bacteria and subsequently cuts them before they can cause any damage. This shows off the bacterial immune system and how it's defensive against foreign DNA, which in this case would be a virus. So, to reinstate my point from earlier, when the tracer RNA is within the Cas9 enzyme, this means that it's guide RNA.
But you might be asking,
so gRNA.
How does the system actually work when DNA gets cut? Well, here's your answer. Once CRISPR-Cas9 makes a cut, the cell has to repair that break to survive. There are two main repair methods. The first is non-homologous end joining, or NHEJ, which quickly sticks the DNA back together, but often makes small mistakes. The second is homology-directed repair, or HDR, which uses a template to fix the DNA more accurately. Scientists take advantage of these two pathways to either disrupt genes or precisely edit them.
Let's think of NHEJ as kicking a soccer ball into your mom's or your vase. You can tell it's been broken and been put back together, but it's not a really good job. Similarly, NHEJ shows where it's been broken and put back together. Let's see some non-homologous end joining in action. Here we have a split piece of DNA, and we've highlighted the PAM site here because that signals where the action is supposed to take place within the cell. Let's focus on some key players, one being COO7080, which is attracted to the ends of the split strands. Then you have the DNA protein kinase, or DNA PK, which is going to stick to the ends of the COO7080 and hold the DNA in place. Let's see some HDR in action. Here you have two homologous pairs of DNA, one of which has been damaged. As a result, RecBCT has come to the rescue, where it receives the DNA until it reaches a Chi site. A Chi site is a combination of nucleotides known as GCT, GGT, GG. Once RecBCT reaches the Chi site, it will stop receiving the DNA, and RecA can do its job, where it will take the DNA to the homologous DNA strand on the other side, and using polymerase, we will repair it. Now you have a fixed strand of DNA, but the issue is it's on the wrong side. So what do? Well, due to complementary base pairing, the strand can return to the original DNA from which it was attained, and you now have a reference from which you can read the process or taking polymerase to fix the strand and ligase to put it all back together. Now you have a brand new piece of DNA on both sides, which is completely repaired and, unlike non-homologous end joining, is entirely correct. Alright, let's have a look at what I just said. Here we have HDR in bacterium, and what we do know here is that you need to have two strands for it to happen. The first strand appears within the actual bacteria, as you can see here, but the second copy of the DNA is made or sort of formed during the bacteria's life, thus allowing for HDR to occur. Lastly, you have DNA ligase, which will, as we know, put the DNA back together using stray nucleotides from the environment around it. Basically, it's a mumbo jumbo of nucleotides that are put back together using DNA ligase.
One real application of CRISPR is treating genetic diseases such as sickle cell disease. Now there's way more, but for now let's stick to the easy ones. Now I assume we all know this, but this condition is caused by a mutation for the gene for hemoglobin, which makes red blood cells become sickle-shaped instead of round. Now, with CRISPR, scientists can edit a patient's stem cells to fix or bypass this mutation, and the corrected cells then produce healthy red blood cells, reducing symptoms and improving overall function. Now this treatment is done by using an ex vivo approach, which means that the cells are edited outside the body. I'm pretty sure the last group talked about this pretty good. Now, first stem cells are extracted from the patient's blood or bone marrow. Then, these cells are sent to a lab where CRISPR is used to edit the faulty gene. Now, in the lab, CRISPR is delivered into stem cells using a method called electroporation, which uses an electric pulse to open tiny pores in the cell membrane, allowing the CAS-9 protein and the guide RNA to enter the cell. Once inside, CAS-9 travels into the nucleus and is guided to a specific DNA sequence. Then, it cuts the DNA, allowing the cell's repair system to modify the gene. Now, before the edited cells are returned, the patient undergoes chemotherapy to clear out existing bone marrow cells and make space for the new ones. Then, the edited stem cells are injected back into the bloodstream. These cells travel to the bone marrow and begin to multiply, and over time they produce red blood cells that contain functional hemoglobin, often increasing fetal hemoglobin levels. This reduces the formation of sickle-shaped cells, improving blood flow and decreasing your symptoms. As a result, patients experience fewer pain crises and a significant improvement in quality of life.